To channel, we are in blood physiology 25th chapter from guidance immunity and allergy. So actually why we are comparing immunity and allergy in these chapters, you can see they both are related. We'll find out that by the end of this video, so you can understand how immunity and allergy are related. So first let us see some of the important terms here. Chemotaxis. In the name you're going to say chemo means chemical, taxis means towards its attraction. So by which the cells can move toward the certain chemicals in the environment. So nothing but in the environment, because of some chemical they attract towards them. That is a chemotaxis. It is nothing but the movement towards those attracted site. And clone. Clone is a group of identical cell which are mainly derived from the common parent cell. From the same common parent cell they are formed, but they form a clone. Means they are, you can say according to humans, for example twins are coming from a same parent, but they are like clones, right? A group of identical cells derived from common parent cell. And next, coming to epitope is mainly an antigenic determinant that is present on a, that is a part of antigen that is recognized by a immune system. So epitope is nothing but an antigenic determinant site that is present in a part of antigen that is mainly recognized by the immune system. Our immune system finds out this epitope which is having an antigen on it. Now let us see what is lysis. Lysis means, of course, we know something is breaking down. So breakdown of the cell by different mechanisms. What are those mechanisms? All those we'll see. So by different mechanisms, if the cell is undergoing breakdown, that is called as lysis. Now let us start with our topic immunity first. Later, next we'll discuss the allergy. So immunity is nothing but the ability to resist all types of organisms or toxins that damage the tissues, organs. So you can understand any type of organism or any type of toxin which is coming to our body, always it will not affect us, so that we are healthy all the time. Only few times we get disease, or only few times we get affected whenever these mechanisms are failed. So whenever you get any kind of disease, you can understand that this immune mechanism is failed in that case. So immunity, it is actually it is divided into acquired and innate. Innate means, in the name you can understand, in and nate. In means inside, nate means from our birth. So innate means which is immunity which is present from our birth, and acquired, you can understand something is acquired, that is after the birth. So this both in when compared with this innate and acquired immunity, the acquired immunity is the most common when compared with the innate immunity. And this acquired immunity mainly develops only after the attack by certain type of bacteria, viruses, toxins. Often weeks or months is required for this immunity to develop. So for example, if you are infected with some of the bacteria, for example with common cold, rhinovirus. So if you are affected for that, again the second time or third time when you affect with that, then only the acquired immunity can be formed. That was I saying. So only after attacked by certain type of toxins, bacteria, virus, then only it will form. That also it takes sometimes weeks or months to require this immunity to develop. Now let us see the innate immunity. Innate immunity example, you can take phagocytosis of bacteria and other invaders by WBC and cells of macrophage system. So destruction of swallowed organisms by acid secretions of stomach and digestive enzymes. So all these are like innate immunity from a birth only. For example, if you swallow some of the toxins, then these are neutralized by the acid secretions from our stomach and by digestive enzymes. And sometimes, by macrophages mainly involved in phagocytosis means eating of the bacteria and other invaders, also by WBC and cells of macrophage system. So resistance of the skin to invasion by organism, even skin as a barriers for this. All this is innate immunity. And lysosome is a mucolytic polysaccharide which attacks the bacteria and cause them to dissolve. Actually this lysosome, it can be released from our tears. Also it is present in our cells. Whenever this phagocyzed vesicle comes near to it, it goes there and just destroy it. So it's a mucolytic polysaccharide, attacks the bacteria and cause them to dissolute. And basic polypeptides which react with and inactivate certain types of gram positive bacteria. So gram positive bacteria are inactivated by basic polypeptides. So all these are like innate immunity, because you can see they're like first grade immunity which are not allowing any organism to enter inside. So whenever your innate immunity is also not proper, then may have the chance of getting the invader inside and get a disease. So the complement complex, it is mainly having the 20 proteins which are activated in various ways to destroy the bacteria. We will see this complement complex later. And also we have the natural killer lymphocytes which recognize and destroy the foreign cells, tumor cells, even some infected cells. And this innate immunity make the human resistant to paralytic means in viral infections, means viral infections which cause paralysis, all those kinds of infections are also resistant mainly because of innate immunity of maybe of animals, or cholera, cattle plague, all this hog cholera, distemper. Usually this distemper virus kills the dogs. So all this kind of diseases, for all these kinds of diseases we are resistant only because of the innate immunity. So animals sometimes, you can say animals are immune to some human diseases, in the case of poliomyelitis it can't affect the animals. Mumps, cholera, measles, syphilis, which are lethal to the humans, but they can resist, they are resisted by the animals. So there are, these are the differences between animal and humans which can resist some diseases of lethal diseases of humans. Now we will move on to acquired. So we have understood innate means which is coming from our birth, and acquired means only after you got affected with some of the disease then only you will get this immunity. But remember that acquired immunity is most common when compared with innate, and also acquired immunity is most best because it's more better because it has memory cells whereas innate don't have. So even if you get affected many times, because of that memory cells which lie in your body for many years, they attack the again that same pathogen or infection. So let us see the acquired immunity. This is also called as adaptive immunity. It is extremely powerful when compared with innate immunity, and it is extremely powerful against the lethal bacteria, virus, toxins, and also foreign tissues from other animals. So this ability is called acquired or adaptive immunity. So this acquired immunity mainly uses antibodies or activated lymphocytes that attack and destroy the specific organisms or toxins. So you can understand, in innate immunity only our barrier system, means which are naturally present in our body, only they get activated and they destroy the invaders. Whereas in the acquired immunity, mainly antibodies and activated T cells play a major role in fighting the and destroying the organisms or toxins. So examples you can take here: paralytic botulinum toxin or tetanizing toxin of tetanus, which mainly protect against the doses high as 1 2 3 4 1 lakh times, that is lethal without the immunity. So remember, paralytic botulinum toxin, tetanus toxin of tetanus protect against doses high as 1 lakh times, that is lethal without immunity. So it is so much lethal that if there is no immunity, they will completely destroy the organism, very lethal. But because of the immunity, we are able to now defend this botulinum toxins or tetanizing toxins of tetanus. So treatment is called as immunization. So treatment for this kind of diseases is immunization, so important to protect against the toxins. So acquired immunity again is further divided into two types. Innate is only one type, that's it. But acquired, as we said, it's the most important, most potent, so for sure it should have some subdivisions also. So the acquired immunity is subdivided into humoral immunity and cell mediated immunity. This humoral immunity is also called as B cell immunity because mainly B cell is involved here, and cell mediated immunity is also called as T cell immunity because T cell is involved here. So humoral immunity mainly produce the antibodies. If you remember, always remember, wherever you find B cell, you can understand that it will convert into plasma cell and it will form the antibody. So humoral immunity mainly produce the antibody, that is globulin molecules in the blood plasma. You know, antibodies are present in plasma, extracellular fluid, right? So which attack the invading agent. Whereas cell-mediated immunity always have the activated T lymphocytes in lymph nodes to destroy the foreign agent. So T lymphocytes, you know, they are trained in the thymus and later they are sent to the lymph nodes. So in lymph nodes, they are always ready to attack the invading organisms. So both antibodies and this activated lymphocytes, which are formed in the lymphoid tissues of the body, protect us. That is mainly acquired immunity. So both types of acquired immunity are activated mainly by antigen. Of course, antigen is required for the case of acquired immunity. Whereas innate immunity, we will not say specifically antigen antibody because it's like a natural process. So whereas in the case of acquired immunity, we will say that antigen is required to activate either T cell immunity or B cell immunity. So antigen is nothing but a protein or a large polysaccharide which initiate the acquired immunity. So this antigen is, yeah, antibody generations. These antigens are also called as antibody generation because when an antigen is formed, then only antibody will form against it. So antigen is having a high molecular weight, like about 8,000 or greater than that, and this antigen is the process which mainly depends on the epitopes. What are epitopes? Epitopes are regularly recurring molecular groups, and this explains why proteins and large polysaccharides are always antigenic because both have stereochemical characteristics. So lymphocytes are mainly responsible for the acquired immunity. So if lymphocytes are destroyed, then there is no acquired immunity because lymphocytes are mainly involved in the acquired immunity. So when there are no lymphocytes, then no acquired immunity. Yeah, and that person dies by many bacterial infections because we are saying acquired is the most potent and most important one. Of course, innate can work, but innate can work only to some extent. Acquired plays a major role. So, if lymphocytes are not present, then acquired immunity is not there, and the person dies with many bacterial infections. So, lymphocytes are most common in lymph nodes, spleen, submucosal area of GIT, that is pairs, patches of ileum, thymus, bone marrow. So, all these areas we can find out the evident lymphocytes. T and B lymphocytes promote the cell mediated immunity or humoral immunity. Mainly T cells we know cell mediated immunity, and B cells we know humoral immunity. So both are mainly derived from the pluripotent hemopoietic stem cells. You already seen in our WBC cells. So first the pluripotent hemostatic stem cells, they later form the multipotent myeloid and lymphoid stem cells we know, that is nothing but forming the common lymphoid progenitor cell, right? So then T cells go to the thymus. This is called, then they are called as T lymphocytes and forming a cell mediated immunity. And humoral immunity, B cells form antibody which is processed in the mid fetal life and in the bone marrow in the late fetal life, and after the birth also still the B cells formation takes place. Now even now in our body B cells formation takes place in the bone marrow itself, but only in the mid fetal life it is taking place in a first in the liver, later into spleen, and finally into bone marrow. So humoral immunity is first discovered in Bursa fabricus in birds, hence the name B. B for Bursa, B for birds, B for B cells, and this mainly form the antibody which from the activated plasma cells. So first B cells convert into activated plasma cells, and those plasma cells later form this antibodies. Now we'll check out the T cells. So T cells first, they are in the bone marrow. From bone marrow they go to the thymus by means of circulation. First from the bone marrow they'll enter the circulation, and after entering the circulation they'll reach the thymus, and again from the thymus they'll move on to the circulation and finally reach the lymph node, and then lymphoid tissues, they stay there. So thymus, one thymic lymphocyte develops specific reactivity against one antigen. Means the every type of cell have specific type of antigen and specificity of antigen. So thymus mainly mix the T lymphocytes with the self specific self antigen, and if reaction is positive then it is destroyed and phagocytosed. So you need to understand this. Self antigens means our own body antigens, and if our T cells find out that their own body antigens, they are destroyed, so that the cells which can't recognize our own body antigens, they are kept there so that they can attack our other cells. That is the reason behind this. So instead of being released in 90% of the cells, so only cells which release and which are not reactive to the self antigen, only these cells are released. Means you can understand, our this T cells should act on the invaders but not on our own cells. But if we find some of the T cells which are in the thymus, only this process takes place. It's nothing but training of the T cells. For example, this is one T cell. If this T cell mix with our self antigen, this is self antigen, right? So the T cell mix with the self cell and reaction takes place. If reaction is positive, then this is phagocytosed and destroyed, because this T cell actually its function is to kill the foreign particles but not our self antigen. So these kind of T cells are destroyed, and only the T cells which are released and left to the circulation to the lymphoid tissues, which are not acting on a self antigen, instead they are acting on the foreign invader. So only these kind of T cells are selected and sent to the lymph nodes. So that is if they act only on the antigen from outside. This occurs before the birth of the baby. So this all process occurs before the birth of the baby and a few months after the birth. So later the T system is decreased due to the regression of thymus. So mainly later in our life only B cells are present. So only till puberty I think these T cells are remain, because thymus regression takes place. And removal of thymus before birth prevent all cell mediated immunity and responsible for transplant organ rejection like heart, kidneys. So all this also takes place if we remove the thymus before birth. And B cells are processed mainly in the liver during the mid fetal life, and later in the bone marrow during late fetal life, and also after the birth. Now also B cells are produced from the bone marrow itself, and these B cells mainly secrete the antibodies. So these antibodies are the reactive agents. These are the large proteins which combine and destroy the antigenic substances. So the greater diversity than B cells mainly have the greater diversity than T cells, thus forming the millions of B lymphocyte antibodies. And B cells also migrate to the lymphoid tissue throughout the body. Here they lodge but slightly removed from T lymphocyte areas. Even B cells are stored in lymph nodes, but mainly T cells are only stored more. And because of overpopulation of T cells, some of the B cells are also removed by the T cells. And different lymphocytes are capable of forming one specific antibody or T cell called clone of lymphocytes are formed. So several hundred types of gene segments as well as millions of different combinations in which segments are arranged in single cells can understand the millions of different gene types and finally form T or B cell. Gene structure codes for only single antigen specificity. And then mature cells, highly specific T or B cells, are spread to populate the lymphoid tissue. So each clone of lymphocyte is responsible for the single type of antigen. And in B lymphocytes on the cell surface membrane, there are about 1 lakh antibodies that will react highly specifically with only one type of antigen. So every type of antibody only acts on one type of antigen. They have some specific antigen specificity. So T cell have the surface receptor proteins or T cell markers which are highly specific for only one specified activating antigen. So antigen stimulates only those cells that have the complementary receptor for the antigen and already committed response to it. So the the macrophages in the tissues mainly line the sinusoids of lymph nodes, spleen, other lymph tissues, and most invading organisms are first phagocytosed and partially digested by the macrophages. And antigen products are liberated into the macrophage cytosol. So then macrophages pass these antigens by the cell to cell directly to the lymphocytes, leading to the activation of specified lymphocytic clones. So in addition, they secrete the special activating substance mainly known as interleukin 1. These are also called as lymphokines. This mainly promote the further growth and reproduction of the specific lymphocytes. So the role of T cells in activation of B cells is so this is how actually what happens whenever invading organisms comes. First the macrophage engulf them and after engulfing them their antigens are released into the macrophage cytoplasm. After they are released, now these antigens are presented to our antigen presenting cells and later this T cells can find out this as antigen and forms a antibody with the help of B cells. So actually T helper 2, we have T cells two types, T helper 1, T helper 2. T helper 2 sends the message to the B cell to produce, get activated to form plasma cell. In turn this plasma cell forms the antibodies. So this is a major mechanism that was saying. So role of T cells in activation of B lymphocytes is actually because antigen stimulates both T cells and B cells, and T cells mainly form the Th cells. These are T helper cells which secrete the specific substance called as lymphokines. These lymphokines in turn activate the B cells. So antigens in macrophages in lymphoid tissue phagocytose antigen and present it to the adjacent B lymphocytes, and antigen presented to the T cells and activate the T helper cells, and extreme activation of B cells takes place. So B cells enlarge and take appearance of a lymphoblast, differentiate to form the plasmablast, which is a precursor of the plasma cell. So the plasmablast cytoplasm expand and its receptors also proliferated. So plasmablasts begin to divide at the rate of about every 10 hours of 9 divisions, giving in 4 days a total population of about like 500 cells per each original plasmablast. So mature plasma cell produce gamma globulin antibody at extremely rapid rate, about like 2000 molecules per second for each plasma cell. So this process continues until the death of the plasma cell takes place. So few clone of B cells don't go to the plasma cells but form a moderate number of new B cells similar to the original clone and circulate, and they are dormant until activated by the same antigen, and these are called as memory cells. So the same cells activated by the same antigen, more potent responses form. So this show why immunization is done by injecting an antigen in a multiple doses which are periods of several weeks or several months between the injections. So this from this we need to understand that actually, we have said that B cells are activated by means of T helpers 2 cells. So this activates the B cell and B cell form the antibodies. Okay, but there are also some other B cells which are activated by the same antigen. They are cloning. I means they are forming identical groups and they are dormant and they are like storing in a cell. They are called as memory cells. So again, this same kind of infection attacks the someone. Then now these memory cells get activated and produce an antibody against it. So immunization is the treatment for this, that is nothing but giving vaccination. So hence you can see that we give vaccination in multiple doses because we can more we can aggregate this kind of memory cells. So all the time this kind of huge memory cells are produced in our body. So if that infection comes again to us, we can easily attack that infection. So this is how we do the immunization process, that is they are explaining. So antibodies are mainly gamma globulins, which are called as immunoglobulins. Its molecular weight is about like 1,60,000 to 9,70,000, and 20% and 20% of the plasma proteins are also involved in this. It also constitutes 20% of the plasma proteins. So remember that even in immunoglobulins, that is our antibodies, we have some plasma proteins 20%, and most of the immunoglobulins are GABA globulins. So till now we have studied about how we attack by means of antibodies. Now we'll see about antibodies in detail. So IgGs, that are immunoglobulins, are mainly the compound of light and heavy polypeptide chains. So most are the combinations of two light chains and two heavy chains. So some immunoglobulins are combination of as many as 10 heavy chains and 10 light chains. Actually normally we have two light chains and two heavy chains. For example, an immunoglobulin looks like this. So here you can see this. So these are the two light chains, two heavy chains. So like this they are arranged, but sometimes there are some big immunoglobulins, like they may also have 10 heavy chains and 10 light chains, which give to rise to increase in molecular weight. Because you can understand, if more amount of chains means more amount of molecular weight is increased. So in each immunoglobulin, the heavy chain is paralleled by a light chain. Yeah, you can see they are parallel to each other at one of its end, and thus forming a heavy light pair, and at least two always, and 10 such pairs in each immunoglobulins molecule is present. So end of light and heavy chain called a uh, variable portion. So we will see this all variable portion in our textbook, right? So it is called as variable portion, and remainder of the chain is called as constant portion. So where this heavy chain and light chain end, that point is called variable portion, and the remainder of the chain is called, means remaining portion is called constant portion. So variable portion is different for each antibody, and this portion attached specifically to particular type of antigen. So constant proportion of antibody determines other properties of antibody, establishing such factors such as antibody diffusivity in tissues, adherence of antibody to specific structures in tissues, and also attachment to complement complex, and ease with antibody passed by the membrane, and other bio properties of antibody, and a combination of non-covalent and covalent bonds, that is like disulfide bonds, and holds the light and heavy chains together. And amino acids organization have different shapes for each antigen specificity. So when antigens come in contact with multiple prosthetic groups of antigen, fit as a mirror image with those of antibody and antigen. So antigen and antibody are mainly held by hydrophobic bonding, water bonding, ionic attractions, van der Waals forces. Also obeys the thermodynamic mass law, like Ka. This is called as affinity constant Ka, and measure how tight the antigen and antibody are bind. So Ka is equal to concentration of bond between antigen and antibody by concentration of antibody into concentration of antigen. So antibody is bivalent, it has two sides for antigen binding, and few have 10 light and 10 heavy chains have as many as 10 binding sites. And